Hello friends, welcome to my YouTube channel. Today we will study about mechanism of cell injury, but before that we need to know about the general principle of cell injury. So let's come to the point, general principle of cell injury. The first is cellular response to injury. Cellular response to injury depends on types of injury, duration of injury and severity of injury. Okay. The second is consequences of injury. It depends on type of cell involved, adaptability of cells, status of cell, and genetic makeup of the cell. So for example, in this one, consequences of cell injury, like in cardiac muscles is highly susceptible to hypoxia, okay? And the cardiac myocyte will die as they are deprived of oxygen. Now the third is targets and biochemical mechanism of cell injury. Like it's had like mitochondrial damage or dysfunctions, disturbance of calcium homeostasis, damage to cellular membranes, and damage to DNA and misfolding proteins. Okay, so these are the general principles of cell injury. So now let's come to the point, mechanism of cell injury. We start mechanism of cell injury. So all this which I have written is an overview mechanism of cell injury. After discussing the overview mechanism of cell injury, we'll discuss in details one by one. Okay, so first let's see the overview. Okay, so all of this can cause cell injury, which are given below. One mechanism is via ATP depletion. In ATP depletion, ATP is less, it will cause a lot of effect. Okay, the second is mitochondrial damage. Mitochondrial damage, which can cause leakage of some pro-apoptotic molecules, some molecules that promote apoptosis, and that will result in cell injury and cell death. Okay, the third is increased calcium entry. It will have a lot of effect by increasing mitochondrial permeability and as well as activating certain enzymes. See, I have written in short like increase mitochondrial permeability, and the second is activate enzymes. Okay. The fourth is ROS, reactive oxygen species. Okay. In reactive oxygen species, it can damage your lipid, protein, and DNA and can cause cell injury, damaging of lipid, protein, and DNA. Now the fifth one is membrane damage. Okay, our cells have a lot of membranes. Say for example, lysosomal cell membrane, and all those membrane can damage. Those can damage. Okay, so the sixth one is misfolded protein and DNA damage. In this, it can promote to apoptosis. Apoptosis, okay, like cell death. It can goes to cell death. So as I have discussed you the overview mechanism of cell injury, let's come to the point, ATP depletion. We'll see ATP depletion, what happens in ATP depletion in details. I have written this short overview, okay, for ATP depletion, but I'll make you understand in details about this. But you need to know this one, what happens in ATP depletion. So come to the point, ATP depletion. In ATP depletion, adenosine triphosphate, ATP, is required for all processes within the cell. Okay, now injury like hypoxia, chemical example, cyanide can cause decreased production of ATP, and major cause of ATP depletions are reduced supply of oxygen and nutrients, mitochondrial damage. This in ATP depletion, I have written in short like consequences or like overview. You can say what happens in ATP depletion. I have written in short point wise. I'll let you know in details what is ATP depletion, what happens actually in ATP depletion. Okay, the first one is effects of decreased ATP. Okay, failure of the cell membrane, potassium and sodium pump, increased anaerobic glycolysis, failure of the calcium pump, and failure of protein synthesis in ribosomes. Okay, so now come to the point, ATP depletion. I'll let you know in details. In details we'll see now. Okay, so to understand this one, ATP depletion, I have drawn a flow chart. Okay, I have drawn a flow chart over here. See the flow chart I have drawn over here, and the summary I have drawn over here, that will make you understand easily. Okay, let's see this chart. Okay, in hypoxia, chemical or toxic injury affects mitochondria and damage mitochondria. Okay, and there will be decreased oxidative phosphorylation, and there will be decreased oxidative phosphorylation, and due to this it will decrease in ATP. Okay, so it decrease in ATP will have three consequences, like cell membrane, anaerobic glycolysis, and failure of calcium pump. Okay, in cell membrane, decreased sodium pump, okay, will have an entry of sodium and efflux of potassium. So, and dilation of endoplasmic reticulum will have swelling cells, loss of microvilli and blebs, and detachment of ribosomes leads to protein synthesis, decreased protein synthesis. In anaerobic glycolysis, in this lactic acid will increase and pH will decrease. When pH will decrease, there will be a chromatin clumping. Okay, and failure of calcium pump, influx of calcium will take place, and activation of damaging enzymes will also take place, and then we'll go to cell death or necrosis. In short, I have written in ATP, anaerobic glycolysis, pH will decrease, and due to decrease of pH, chromatin coupling will take place. In cell membrane, decreased potassium pump will take place, and then dilation of cell membranes, swells, endoplasmic reticulum swells, and microvilli loss, and detachments of ribosome takes place. So now I have drawn this chart, and you have seen this chart. I'll explain you in more details about it. So when let's, for example, okay, for example, there was ischemia in blood vessels. Okay, that means blood was reduced in a blood vessels. It may be due to some thrombus or some other occlusion. So when blood supply is reduced to that tissue, what happening there? Oxygen supply is getting low. So that is known as hypoxia. Hypoxia means reduced amount of oxygen in the tissue. Then what happens when there is hypoxia? The electron transport chain cannot function properly because oxygen was used as an electron acceptors in the electron transport chain. Okay, so there will be loss of ATP synthesis from the electron transport chain, and when there will be a less ATP synthesis, and that will cause mainly three phenomena. When there will be a loss of less of ATP synthesis, it will have three mainly phenomenon. Okay, the first thing is we need ATP, we need energy to survive, so when there is absence of oxygen, the cell of our body will try to generate ATP without oxygen. Not much, but in little amount, it will try to generate some oxygen, and that process is known as anaerobic glycolysis or anaerobic respiration. In this process, it will make some product, and that byproduct, okay, and that byproduct is known as lactic acidosis or lactic acid. And so when there is acid in the environment, the pH will drop, and as a result of the drop in the pH, the chromatin material inside the cell will begin to clump. So that is the mechanism of ATP depletion. Okay, ATP depletion. So different when there is ATP depletion uh, in cell injury, different pumps in our body will not function properly. Okay, one of this important pump is sodium pump, and the function of sodium pump was to pump sodium out in exchange of potassium. Okay, so now since we have less ATP, that pump will not work properly. Okay, and as a result sodium pump, that means sodium pump will not function properly, and as a result what will happen, sodium will try to accumulate, and that will result in cell swelling. Okay, when sodium will accumulate, there will be accumulation in cell. Okay, cell means like cell swelling will take place. Similar mechanism will also seen in endoplasmic reticulum, that will result in endoplasmic swelling. And there will be one more thing, when the cells are getting swelled up. Okay, when the cells are getting swelled up, a lot of cell has microvilli. Okay, when the cells are getting swelled up, a lot of cell has microvilli. I have drawn over here. Microvilli is a finger-like projection. Okay, so when microvilli is a finger-like projection, so when the cell is getting swelled up, okay, when the cell is getting swelled up, those things will get lost. So there will be this finger-like position. When cell gets swelled up, this finger-like position will be get lost, or will be lost because of cell injury. Means like cell swell up, it will get loss of microvilli will take place. Okay, so now come to the point, detachment of microsomes. Okay, now come to the point, detachment of microsome. So microsome, the last mechanism by which ATP depletion can cause cell injuries, when the cell endoplasmic reticulum is getting swelled up. Okay, if you remember, like, what is rough endoplasmic reticulum? Rough endoplasmic reticulum, those are which ribosomes are attached with it. Okay, so when those rough endoplasmic reticulum are swelling up, what will happen? The ribosomes which were attached to those endoplasmic reticulum, they will detach. And since ribosomes is made up of ribosomes is since ribosome is the site of protein synthesis, so there will be reduced amount of protein synthesis. Look here, I have drawn. The last thing that will happen due to ATP depletion is the detachment of ribosomes, and we know that ribosomes are more important for our protein synthesis. So when the ribosomes are getting detached from their site, it will cause decreased amount of protein synthesis. Okay, so let's come to the point now. I have discussed ATP reduced pumps will fail. So what I have discussed, pumping also have discussed, and detachment of ribosomes I have also discussed. Let's come to the point, mitochondrial damage. Okay, what is mitochondrial damage? This is an overview which I have written over here. Okay, the second mechanism, it is a second mechanism by which mitochondrial damage takes place. Mitochondrial damage may be viewed as mini factories that produce life-sustaining energy in the form of ATP. Okay, mitochondria are sensitive to many types of injuries, stimuli, including hypoxia, chemical toxins, and radiation. Now, these are the consequences of mitochondrial damage. See, the first one was ATP depletion. Second is formation of reactive oxygen species, and the third is formation of mitochondrial permeability transition pore. In this, it occurs in mitochondrial membrane. Okay, this leads to the loss of mitochondrial membrane potential, pH changes, and progressive depletion of ATP, and ultimately it leads to necrosis, okay, of the cell. C. And the fourth is leakage of mitochondrial proteins in the cytoplasm. This what I'm reading, I have written in short and brief, so you can just mug up or just understand, and I'll make you understand in details over here. Okay, and the fourth is leakage of mitochondrial proteins into cytoplasm. The mitochondrial membrane contains many proteins such as cytochrome C and pro-apoptotic proteins. Example BAX. Okay. So increased permeability of the mitochondrial membrane may result in the leakage of these proteins into cytosols and induce apoptosis. See, I have drawn over here the structure of mitochondria. Okay. So now I'll explain you in what leads to mitochondrial means mitochondrial leakage or mitochondrial damage, what happens in this. Okay, first of all, what is like functions of mitochondria? There is always remember, like mitochondria is also known as powerhouse of the cell. The energy that is needed for all the function of the cell is the in the ATP, like the energy that is generated for all the function of the cell in the ATP is generated on the inner membrane. Okay, it generated on the inner membrane. In mitochondria, it has inner membrane. The ATP is generated over here, like in the inner membrane, and it also has an outer membrane. See, this one is the inner membrane and this one is the outer membrane. So in the inner membrane, those ATPs are generated via electron transport chain and oxidative phosphorylation. Okay, so whenever there is mitochondrial damage, one thing will happen. One thing will happen for surely, like ATP production will get reduced. Okay, ATP production will get reduced, and other things that happen when there is mitochondrial damage. There is mitochondrial damage. Always remember, like important micro organ, always remember, when mitochondria are getting damaged, there is a formation of high conductance channel. Okay, high conductance channel, and that high conductance channel is known as mitochondrial permeability transition pore. Okay, so when mitochondrial permeability is produced, when mitochondria are damaged, and what happens when MPTP is produced? MPTP means mitochondrial permeability transition pore. Okay, is produced. The permeability of the mitochondrial membrane gets increased abnormally. Okay, and as a result, mitochondria in an organelle, and all the organelle has a special membrane potential. So, and the membrane potential will be lost because of mitochondrial permeability transition pore. Because of mitochondrial MPTP, the membrane potential will be lost. Okay, so when the mitochondria will lose its membrane potential, and as a result it won't be able to produce sufficient amount of ATP. So it is the first mechanism by which mitochondrial can cause cell injury. Now let's come to the second mechanism by which how mitochondria uh, can damage or can cause cell injury, is via apoptosis. Okay, always remember one thing, like between the inner membrane and the outer membrane of the mitochondria. See, inner membrane and outer membrane, in between this, in between outer membrane and inner membrane of the mitochondria, there is a lot of proteins. See, this one is the proteins which I have drawn in the black dots. This one is the proteins. In between outer membrane and inner membrane of the mitochondria, there is a proteins, a lot of protein. I have drawn, see, I have drawn over here with the black dots. Okay, and they can activate an important enzymes. Okay, they can activate an important enzymes, an important enzyme known as caspase, which are important for apoptosis. Okay, uh, mitochondria, in mitochondria I have discussed previously, like it will most probably it will have take two place, can cause two things, either activating of enzyme leads to apoptosis, or like the first one will have mitochondrial damage. Okay, mitochondrial permeability. If mitochondrial permeability transition pore will be there, so membrane potential will be lost. Okay. Say for example, there is a protein named cytochrome C. I have said you like mitochondrial membrane contain many proteins such as cytochrome C and pro-apoptotic proteins, example BAX and BAX. There is a protein named cytochrome C. When mitochondria is damaged, when mitochondrial permeability transition pore are present, okay, this protein which I have previously shown you like in black dots, this protein what will do, that were present between inner membrane and outer membrane mitochondria, these proteins may leak out of the mitochondria and they may come in the cytosol, and they can stimulate certain enzymes, and there they can stimulate certain enzymes, and that can result in a special type of cell that is known as apoptosis, which is also known as programmed cells death or suicide of the cell. So it was a mechanism by which mitochondria can damage, or mitochondrial damage can cause cell injury. Now the third mechanism for cell injury was influx of calcium and loss of calcium homeostasis. It is also we will also call as like increased cytosolic calcium. Okay, so the next mechanism let's discuss over here is, we need to always remember that calcium is an important part in this. Okay, so calcium is an important mediatory for all cell injury. Okay, and to prevent cell injury, the calcium level inside the cytosol of the cell is always kept extremely low. Okay, less than like 0.1 meter micromole as compared to 1.13 millimole level of calcium outside the cell, and calcium level is kept low inside. Okay, the cytosol by sequestering calcium inside mitochondria and also inside endoplasmic reticulum, the sarcoplasmic reticulum. So what happened when a cell is injured, say via like ischemia or by some toxic some toxin, though those calcium that were sequestered inside the endoplasmic reticulum and mitochondria, they get released. Also later, when the cell is severely injured, there is some pump failure. So calcium, there is some pump failure. So calcium from outside also get inside the cell. There is also influx of calcium, but but that happens later. Okay, initially after being exposed to ischemia or after being exposed to toxin, calcium level increases inside the cell as a result of release of calcium that was sequestered inside the mitochondria and also inside the endoplasmic reticulum. Later, calcium from outside and also influx and causes high level of calcium inside the cell. So what will happen? Two things will happen over here. This I have written in memorize for this one. Two things will happen over here. One thing is this: high level of calcium will increase in the mitochondrial permeability transition pore and result in cell injury. Okay, now remember that there are some other causes that causes mitochondrial damage. Besides high cytosolic calcium, there are release active oxygen species and hypoxia. So always remember what are the causes of mitochondria damage. Always remember there will be increased cytosol calcium and reactive oxygen species, and hypoxia is the main cause for mitochondrial damage. So I have discussed about influx of calcium and loss of calcium homeostasis. In this, you need to always remember that there will be an increased cytosolic calcium. Okay, increase of calcium production will take place in this. Okay, see in this. So now come to the point. So now what will I have said you, like, note one thing, what are the causes of mitochondrial damage? Okay, so the mitochondrial damage in are causes like increased cytosolic calcium, reactive oxygen species, and hypoxia. Okay, so now we come to know the mechanism of cell injury by calcium injury. Okay, by calcium injury, which second way calcium can cause cell injury is by activating different types of enzymes and causing cell injury. Okay, now uh, what are the enzymes that are activated by calcium? Okay, the first one is ATPase, and it will break ATP and result in second ATP depletion. Okay, the first one is ATPase, and it will break ATP and result in ATP depletion. Okay, the second enzyme is activated by high level of calcium is phospholipase, and we know that phospholipid is an important component of our cell membrane, so that will result in membrane breakdown. The third enzyme activated by calcium is protease. Okay, and the name is easy to understand, like this enzyme will break down different types of proteins inside our cells and causes cell injury. And the fourth enzymes activated by high level of calcium is endonuclease, that will remain that will result in DNA damage. Okay, if the endonuclease if there is a high level of calcium in endonuclease, okay, so there will be a damage of DNA. So this is the mechanism by which cell injury occurs, is by increased calcium entry. Okay, so now come to the point, fourth mechanism, ROS, reactive oxygen species. Okay, this is also known as like oxygen derived free radicals. We also call them as oxygen derived free radicals or oxidative stress. The reactive species are oxygen derived free radicals, and when you will ask what is free radicals, so I have written over here like what is free radicals. Free radicals are a special type of molecules that have unpaired electron in their outer orbit. Okay, there will be an unpaired electron in the outer orbit. So the criteria of calling a molecule a free radical is that has to have a single unpaired electron in their outer orbit. Okay, this one, if there is a single unpaired electron in the outer orbit, then there will be a, we call as free radicals. And the second will be, it should be unstable. The structure should be unstable, not stable. Okay, and therefore the free radical will always tend to react with whatever they come in contact with, that can be carbohydrate, so proteins, extra. So this is free radicals and they can cause cell injury. One important we have to remember for like free radicals is they can be autocatalytic reaction. Okay, they can be autocatalytic reaction when the free radicals is reacting with carbohydrate, proteins, or lipids, they can convert those carbohydrate, lipids, and proteins into free radicals themselves. That will that will result in chain reaction. Okay, so now we'll discuss the mechanism of free radicals formation. The causes of free radical formation is, there are five main causes by which free radicals can be generated. Okay, the first cause of free radical generation is via the normal reduction and oxidation reaction. Normal reduction and oxidation reaction is redox reaction that occurs during different metabolic process inside our cells during molecular respiration. Okay, to understand this better, I have written in like in brief or in simple words, is like, for example, oxygen During this molecular respiratory process, oxygen get reduced and transfer 4 electron to hydrogen and forms 2 molecule of water. Okay, 4 electron of to hydrogen and forms 4 molecules of water. So that is a normal reaction. By sometime there is partially reduction of oxygen and that will result in formation of free radicals. For example, when 1 electron is transported, there will be formation of a free radicals, which is known as superoxide anion. Okay, superoxide anion. When 1 electron is transported, there will be formation of free radical, which is from out of 4, 1 electron is transported, then uh, there will be formation of free radicals, which is known as superoxide anion. So this is due to transfer of 1 electron instead of 4. Okay. Now the second free radical produced is hydrogen peroxide, H2O2. Okay, hydrogen peroxide, hydrogen peroxide is the second free radicals produced. Okay, then here there is transfer of 2 electron instead of 4. Out of 4 electrons, there will be only transfer of 2 electrons, so hence named hydrogen peroxide. Okay. Third free radical is hydroxide ion. Okay, hydroxide ion OH, due to transfer of 3 electron instead of 4. So all these are produced due to partial reduction. Okay, but normally these things are produced in a small amount and the cells have some antioxidant. Okay, some antioxidant. So antioxidants, say for example, like vitamin A, E, or C. For remembering vitamin A or C, in this it has a lot of antioxidants. You can remember as ACE. Okay, there are antioxidants. They can neutralize those free radicals. Okay, those free radicals from those free radicals when they are on a small number. Okay, and when they cannot neutralize due to some reaction, there is high amount of free radicals and that result in a term known as oxidative stress. Okay, the second cause is first you should know about what is antioxidant. What is antioxidant? Okay, so antioxidant, you can say are compounds that inhibit oxidation. Okay, so in this, oxidation is a chemical reaction that can produce free radicals. Okay, hence leading to chain reactions that may damage the cell of organisms. Okay, now, so antioxidants such as thiols or ascorbic acids terminates this chain reaction, as I have said you here, like vitamin A, C, or E. Okay. The second cause by which free radical is produced is via ionizing radiation or absorptions of radiant energy. Okay. That can be due to X-rays or UV rays, etc. Okay. Third mechanism by which free radicals can be produced is occurs during inflammation. Inflammation or rapid burst of reactive oxygen species during inflammation. Okay, during let's see like what happens in this. Okay, during inflammation, some cells in our body can make reactive oxygen species in high amount, which is a term known as uh, reactive oxygen species burst. Okay, so and they use those ROS species, mainly H2O2. Okay, hydrogen peroxide and HOCl minus. Okay, which is an potent antibacterial agent to destroy harmful organisms. So the fourth cause of this is forming free radicals is during metabolism of some exogenous molecules. For example, carbon tetrachloride During its metabolic process, it produces carbon trichloride, CCl3, and that is a potent free radical. Okay. The fifth cause is transition metals. In transition metals, which includes iron and also copper, and they can produce free radicals. Okay. So one thing you should always remember, like there is an important reaction called Fenton reaction that occur inside our cells. I have written over here the reaction. See, H2 hydrogen peroxide plus ferrous will give ferric acid and hydroxide, OH plus hydroxyl ions. Okay. The most of the iron in our body is ferric form, not in the ferrous form. Always remember this one. Okay. So now we'll know the mechanism of cell injury by free radicals formation. Okay, free radicals formation. The reactive oxygen species are free radicals that can cause three problems. Okay, ROS species are free radicals that can cause three problems. The first thing is that they can oxidize lipid, which also known as lipid peroxidation. Example, we know that in our cell membrane we have a lot of lipids, and what happens when those lipids are oxidized? They form peroxidase, and those peroxidase containing cell membrane, they get disrupted. So this is the mechanism of lipid peroxidation, and that will disrupt the membrane. And also remember, lipid membrane lipid membrane. Also remember, lipid membrane not only found in the cell membrane but also found on different organelles, so all those things will also get affected. Okay, so the second mechanism is by which free radical can cause is protein oxidation. Protein oxidation, जैसे कि for example you can see enzymes are also a protein, but all proteins are not enzyme, remember this. So whenever some enzymes are oxidized, they will lose their enzymatic activity, and sometimes there will be abnormal folding, and that will result in cell injury. Okay, so the last mechanism by which reactive oxygen species can cause cell injury is by DNA damage. So they will cause mutation in the DNA, and due to oxidation, and when this oxidized mutated DNA breaks down, that will cause cell injury. So that was the mechanism by which cell injury of reactive oxygen species takes place. Now the point last but not the least, cell damage. How cell damage takes place? What are the mechanisms of this membrane damage? Membrane damage takes place. Okay, membrane. Remember, we have membrane in our cells, which is the cell membrane, and also in different organelles, and they can be damaged. Okay, what are the most common causes of membrane damage? Will be like we have already discussed so many causes, but important cause is hypoxia. Okay, whenever there are decreased amount of oxygen in the tissue, there will be ATP depletion, and we have already discussed before, and there is ATP depletion that will result in decreased phospholipid synthesis. Okay, and that will result in membrane damage. The first cause one, this one. Now come to the second cause. The second cause is mechanism of membrane damage via increased calcium in the cytosols. Okay, or increased level of calcium. Let's see, there is a lot of effect in case of mitochondria that used to cause formation of mitochondrial permeability transition for MTPT. Okay, and that used to result in mitochondrial damage. Okay, another thing, the increased calcium in the cytosol it was activated different enzymes. Say for example, like phospholipase, protease, endonuclease, and ATPase. And among the four enzymes, the first two, phospholipase and protease, are responsible for membrane damage, which I have discussed in earlier in mitochondrial damage. Okay, so now let us see how it is damaged, the enzyme, how it is damaged. Okay, so the first phospholipase will break down into phospholipid. That is the important component of the membrane, and that will result in membrane damage. But how does protease cause membrane damage? Okay, protease breaks down cytoskeletal protein, and if you know the function of cytoskeletal protein, the function of cytoskeletal protein is to act as anchoring molecule, and they anchors the cell membrane or other membrane with the cell itself. Means it controls the cell molecule or cell membrane inside or other membrane with the cell itself. Okay, so whenever we are having protease causing cytoskeletal protein damage, those anchoring molecule will get reduced in the number, and the cell membrane will become detached from the cells, as we have seen ischemia in case of myocardial cells, and cause those cells and myocardial cells. Okay, and when those cells get swelled, that will cause rupture of the cell membrane or swelling of the cell membrane. Okay, so those were the causes of membrane damage. uh, Hypoxia and increased cytosolic calcium. Okay, now there are some other causes of membrane direct damage, includes like bacterial toxin, viral proteins, lytic complement component, etc. And even reactive oxygen species can also cause membrane damage. Okay, now come to the last cause. Okay, misfolded protein and DNA damage. Okay, cell injury is caused by misfolded proteins and DNA damage. Now recall that the proteins are synthesized in the ribosomes. Okay, the proteins are synthesized in the ribosomes, but after being synthesized, they fold themselves in different, like three-dimensional architecture, in different, or they fold themselves in structure of like 3D, three-dimensional architecture. It folds in 3D structure. Okay, the protein being synthesized, they fold themselves in 3D structure, in shape of 3D structure. Okay, but sometimes those folding occurs abnormally, either due to some problem in the genes responsible for those protein. So what will happen, those misfolded proteins continuing cells cannot function properly. Okay, so they cannot function properly, and they will get injured. Okay, and after injuring, ultimately they will die. So, and sometimes there will be a mutation in those cells, and sometimes there will be damage to DNA due to some exogenous regions. And what will happen, the cells that containing those damaged DNA, they will die via a programmed cell death mechanism known as apoptosis. Apoptosis leads to cell death. So friends, I have discussed the mechanism of cell injury. So cell injury topic is completed, and hope you find useful this video. If you like this, if you have like means get like this video, so please comment over here and subscribe my channel. So thank you so much.